It's my great pleasure to introduce our keynote and last keynote speaker. So Alice Ting was an undergraduate at Harvard, where she worked with Nobel laureate E.J. Corey. She went to do her PhD with Peter Schultz at Cal Berkeley. And then she did a postdoc at UCSD with another Nobel laureate, Roger Tseng. Alice started her lab in 2002 and then moved she started at MIT in 2002 and then moved to Stanford in 2016. And we have insider knowledge that when she moved her lab to Stanford, they were doing experiments on the day they moved into Stanford.
Within a week.
Within a week?
Yeah.
Okay, so our insider knowledge might be a little limited. All right. So she has many honors. I'll just list a few. She has won the Arthur C. Cope Scholar Award from ACS. She's NIH Director Pioneer Award, Sloan Foundation Research Fellowship, 2 times NIH Transformative Award, and she recently was elected into a member of the National Academy of Sciences. The Ting Lab. Their work in the field of protein engineering is regularly transformative, making different editors, labelers, and recorders. I actually met Alice, I'm not sure if you know this or remember this. I met Alice at a Keystone meeting in Breckenridge in 2017. And she gave probably the most phenomenal talk I have ever seen. This was early on in optimizing BioID, and this is when labeling it took hours to do the labeling, and she essentially transformed the field by bringing that labeling down to essentially minutes. And then she went on and essentially gave a whole other beautiful story of how do you get selectivity and subcellular localization to the labeling. It was absolutely incredible. I was looking around at other people, and their jaws were on the floor, and they were all looking up and saying,
"Oh my goodness!
Oh my God!"
Everyone's expectations are now out of control.
So anyway, it was a fantastic talk and extraordinarily transformative for the field. And so we're really excited again to welcome Alice Ting.
Thank you so much. I'll use this last one. But oh my gosh, that's high expectations. But thank you so much for the incredibly kind introduction. I'm so happy to have the chance to share our recent work with you here. While listening to the talks at this meeting, I've been completely fascinated, and the reason being that our lab has been, I've been working on protein engineering for almost 3 decades, and we've produced various protein-based technologies like Turbo, Apex, calcium integrators, and so on. But our long-standing approach has been, number 1, to start from building blocks from nature, and number 2, to use more traditional methods, most of all directed evolution, to try to generate useful tools. But in recent years, we've really embraced computational design, and so in today's talk, I want to share with you some of our successes in using computational design to engineer proteins, but also our failures, with the hope that those examples might inspire some of you, especially the young scientists who are interested in tackling difficult problems and looking for opportunities. So for this project, it's been carried out by 3 incredibly talented trainees, 2 of whom are here. So Andrew had his poster on Monday, and Peter will be presenting his poster on this work tonight. So please visit and say hello to them. Okay, so what is the reason for developing these enzymes for proximity labeling? So the motivation is that 2 of the most commonly asked questions in biology are, what's in my cell compartment or my organelle of interest? Can I get an unbiased parts list of all the proteins, RNA, metabolites that are there? And secondly, what's interacting with my protein of interest, right? What's its interactome? And the traditional methods for answering these 2 questions that come up all the time are kind of inadequate. So for mapping subcompartments, the standard method is biochemical fractionation, but that doesn't work for a lot of the types of compartments that people are becoming increasingly interested in, like membraneless organelles or the synaptic cleft, the space in between two membranes. There's no way to biochemically enrich that. And so they're kind of unexplored black boxes. And then for mapping interactomes, the predominant method is also a biochemical enrichment. So bait of interest, pull it down, see what comes down with it. The problem is that just the act of cell lysis and detergent and dilution, followed by the washing steps, you lose a lot of real interactors that might be only weakly or transiently associated, but are functionally very relevant. And so we developed proximity labeling as a way to bypass the need for purification or enrichment. And the way it works is that you take an engineered promiscuous enzyme and you genetically target it to either your organelle of interest or your protein complex of interest, just by fusing it to a localization peptide or fusing it to your bait protein of interest. And then you supply to the cells a small molecule substrate, like biotin in the case of turbo-ID or biotin phenol in the case of Apex, that by itself is not reactive, but when it encounters the active site of these enzymes, it gets converted into something very reactive and short-lived, that like a adenylate ester or a phenoxyl radical that diffuses out of the active site and covalently attaches nearby endogenous proteins in a sequence-independent way, so promiscuity. And the labeling radius is just on the scale of a few nanometers, and so you can get really high spatial specificity with this kind of spray painting, labeling that goes on. Importantly, in intact live cells, while membranes, complexes are all intact. And after you perform that labeling, for just 1 to 10 minutes, then you lyse your sample, you harvest your spray-painted proteins using streptavidin beads, for example, and then you identify what you have by mass spec. So these enzymes have been now widely used to map organelle proteomes and subcompartment proteomes, and also interactomes. However, they do have important limitations, and so our lab is interested in continually improving these technologies so that they can be more useful. So for Apex, for example, a major limitation is that it's a peroxidase, and so it requires hydrogen peroxide, which is toxic, so people don't want to use it in vivo for proximity labeling. And then for turbo-ID, the main problem is that it has a strict requirement for biotin. And all living organisms use biotin and have biotin-related proteins, so that's a source of background. How do you distinguish what was labeled by turbo-ID from the background that was already there? And so we've been interested in developing a new enzyme, which we're calling FlexID, that can use diverse non-biotin substrates and also catalyze labeling under non-toxic conditions. Okay, so this is the motivation. And the starting template, so now we're kind of going to our playbook where we look for some natural enzyme in nature that we can start engineering from. And for this, we picked this E. coli ligase called LPLA for 2 reasons. Number one, it's homologous to Bure, which was the template for turbo-ID. So like Bure, it catalyzes ATP-dependent ligation of a cofactor, lipoic acid, onto the lysine side chain of an acceptor protein. The second reason that it's an attractive template is that we previously found that you can take this tryptophan side chain that's at the end of this lipoic binding tunnel, and if you mutate it to valine and just make this area roomier, then this point mutant can accept diverse small molecule alternatives to lipoic acid. So it can ligate fluorophores, it can ligate these click chemistry handles, which are these bioorthogonal functional groups that enable facile enrichment of labeled proteins. So that had already been established, and as a shout out to David, actually LPLA is a template that we collaborated with his lab about 10 years ago. Lucas Nivan in the Baker lab designed this triple mutant of LPLA that could ligate this red fluorophore in place of lipoic acid. And I really love this project because we solved the crystal structure of that resirufin ligase, and there was an excellent match to the computationally predicted structure. Okay, the main problem with LPLA, the main challenge, engineering challenge, is that it's really sequence specific. So in E. coli, it has only 3 substrates. If you express it in mammalian cells, which is the main context in which we want to use these tools, it doesn't label anything. So it's really sequence specific, and we have to make it totally promiscuous. So it's like kind of a big gap. What we could have done, and what we did in the past with Apex and TurboID to address these kinds of engineering challenges was directed evolution, right? So we could have done yeast display and selected for promiscuous variants, but it was a new era. And Peter, my student, to his great credit, really wanted to try computational design. And so he wanted to try 2 different approaches using 2 different models. So one, a sequence trained model, and secondly, a structure trained model. And what he did was he input either the sequence or the structure of the wild type enzyme, and he asked these models to predict point mutations that were plausible or high scoring, right, using ESM or protein MPNN. And then he introduced these point mutations one by one onto the W37 template, W37V template, and we use that because it can utilize this unnatural probe that makes it easy to detect labeling and quantify promiscuous activity, express these mutants inside hex cells, and then measure both promiscuous labeling and expression level. And what Peter found was that protein MPNN was really good at improving enzyme expression. And both models predicted mutants that could increase promiscuous activity, but ESM in particular found 2 mutations that dramatically increased promiscuous activity. And if you look at where these are in the structure of LPLA, they're 200 amino acids apart in the linear sequence, but they're right next to each other in the 3D structure. And where they map to is this hinge region between the C-terminal domain of the enzyme and the N-terminal domain that undergo this large conformational change during the first half reaction that this enzyme catalyzes. And so just that positioning made us wonder, could they be affecting conformational dynamics, right, because they're right in that hinge region, and could that change in conformational dynamics somehow, we don't know how, be causing the enzyme to become more promiscuous, right? So this is what we know about the enzyme from the crystal structures. So we know that the enzyme starts out in this closed conformation with that C-terminal domain downward, and that conformation is necessary to catalyze that adenylation step, right? So activating this acid into an ester, that's the first half. But then, for the second half reaction to occur, which is the transfer, there has to be a conformational change from this closed shape to this open shape, where the C-terminal domain flips up and gets out of the way, because otherwise, there's no room for the acceptor protein to come in and dock, right? So it has to kind of be like a transformer robot that changes from closed to open in order to catalyze the second half reaction, right? So in this open conformation, the acceptor protein can come up, reach its lysine into the active site, attack this activated ester, and form an amide bond, right? And that completes the transfer step. But we know that crystallography is not always an accurate representation of what happens in solution, right? And if we want to study conformational change, we needed a solution measurement of what was going on, right? Crystallography, it's a purifying process, right? It's a purification. So we selected small angle X-ray scattering as a solution measurement of conformational change, because we kind of didn't have many other choices. So LPLA at 35 kilodaltons is too big for NMR, too small for cryo-EM. Thankfully, the conformational change is large and easy to detect by SACS, so that's what we used. So first we did measurements on the template, the W37V, and we found that in the APO form, it's almost entirely closed. That matches what we see in the crystal structure. But in the holo form, when we give it lipole AMP ester, right, the adenylate, it doesn't fully open. It only partially opens. And that's in contrast to what the crystal shows, right? Interestingly, if we measure this ESM predicted point mutant that was much more promiscuous, right, than the template, it starts out also completely closed, but then when we supply the adenylate ester, it becomes entirely open. So it was true. That point mutation is dramatically altering conformational occupancy, right, of this enzyme in the holo form. And we also used a tryptophan fluorescence assay, which led us to the same conclusion, okay? And this is just a different way of looking at that conformational change. Okay, so, so that was very interesting. And just now seeing a correlation between conformational occupancy changes and promiscuous activity made us wonder if we could now deliberately design for open conformation bias and generate even better enzymes for promiscuous labeling that way, right? So ESM, we were just lucky. I mean, we weren't able to use ESM in a way where we could specify that our objective was to make the enzyme more promiscuous, right? But now we have a hypothesis about how conformation is related to promiscuity. And so Andrew and Peter came up with this clever but very simple idea of using protein MPNN, or other inverse folding models work as well, to do contrastive scoring of point mutants of LPLA against both open and closed backbone structures, right? So the idea is to go through every possible point mutant of LPLA, pair each mutant with both the closed and open backbone structure, use protein MPNN to score it, right? And then you have these pairs of sequence log likelihoods, right, based on both backbone structures. And if you plot these here in a scatter plot against the conformation 1 score versus the conformation 2 score, the things that are off the diagonal are predicted to be conformation 1 or conformation 2 biased, right? And so we applied that to LPLA, right? So we processed every point mutant of LPLA. The vast majority are in the middle. They're not predicted to be biased towards the open or the closed conformation, but there are some that are up here that have a higher closed score than open score, and vice versa down here. And so we took about 100 of these point mutants, and we tested them experimentally, right, to see if our predictions would hold. The easiest assay for us to do is the assay for promiscuous labeling activity, which we're using kind of as a proxy for conformational bias. And I showed you the results of this assay before, but this is how it works. We express the enzyme in the cytosol of HEC cells. We do labeling with this, and we do click chemistry with this. And when we do flow cytometry on the cells, if the expression normalized Bodippy intensity is shifted to the right, it's high, that indicates that the enzyme is more promiscuous. It's catalyzed more labeling of endogenous mammalian proteins with this BCN substrate, and we expect that for the open biased mutants. And for the closed biased mutants, we expect it to be less promiscuous. And so here are the results. The template is down here, W37V. It has a little bit of promiscuous activity. The open biased mutants, the majority of them were more promiscuous, had more promiscuous labeling activity than the template. Some of them dramatically more, right? The closed biased mutants, almost all of them were less promiscuous than the template. And the neutral variants were in between. So this was pretty cool and generally matched our expectations. And for these closed mutants, we wanted to exclude the possibility that they were just inactive and dead. So we also tested a subset of these mutants just with the specific acceptor protein. It's natural acceptor protein, and they all have activity. So it's not that they're dead, they just have lower promiscuous activity, which is important to distinguish. We also took a subset of them, and we actually measured their conformational occupancy, right? It's because the promiscuous activity is just a proxy for what we're actually trying to design using this conformational biasing approach. And what we could see is that the CB predicted open biased variants generally had a greater fraction in the open conformation compared to template, whereas the predicted closed biased variants had a lower fraction in the open conformation compared to template, right? And tryptophan fluorescence, same kind of trend. We wondered, are we really tuning the equilibrium between two conformational states, right, with these point mutants that are giving the results that we're seeing? And so actually one of the reviewers of our manuscript made a really clever suggestion. And they said, what would happen if you combine these biasing mutations and make double mutants in the same protein, right? So if you're actually, if they're acting on the same conformational equilibrium, then you should go back to neutral, right? So what we did is we combined closed biasing mutations with open biasing mutations, and this is the prediction of where their CB scores will be, right, for the double mutants, so it goes back to the center. And so we made those, and we tested them by SACs and by tryptophan fluorescence, and for the most part, we see that these double mutants have conformational occupancies in between that of the parental single mutants. So based on these results, we have two conclusions. One is that CB seems to bias the conformational equilibrium of this enzyme in the adenylate-bound holo state, but not in the apo state, and that open bias variants of LPLA are more promiscuous, whereas closed bias variants seem to be less promiscuous. The question, of course, is why, right? Why is there this conformational gating of substrate selectivity? And so this is kind of our best mechanistic hypothesis right now to try to explain this, because this was kind of a feature of LPLA that wasn't recognized before until we started playing with it, right, and engineering it. So what we think is happening is that for the wild type enzyme and also for closed bias mutants, so after you do the first half reaction, so the adenylation, you have this complex that's in equilibrium between the closed and the open conformation, but for the wild type and closed bias, it's predominantly closed. And what we think is happening is that C-terminal domain is kind of like a tethered competitive inhibitor of the transfer step, right? As long as it's down, you can't do transfer. So only good substrates that have high affinity for this enzyme can capture this minority conformation and proceed to transfer, right? Whereas bad substrates, the off-target proteins, cannot. In contrast, the open biased mutants of LPLA, right here, that C-terminal domain, after it forms the adenylate ester, that C-terminal domain is out of the way. So it's not inhibited. There is no competitive inhibition. And so that active site is available for both good and bad substrates to come in, attack the electrophile, and complete the transfer step, right? So that's kind of our explanation for how conformational equilibrium could tune enzyme selectivity. And I'm really intrigued by the possibility that this might apply to other enzymes as well and not yet be recognized. Okay, so we took these things that we learned from engineering and we made FlexID, but before I do that, let me do a quick detour about this conformational biasing methodology, which has proven useful for LPLA. So we see, this is just the CB bias score plotted against our 3 experimental measurements, right? The promiscuous activity, the tryptophan fluorescence measurement of conformational occupancy, and then our SACs measurement of conformational occupancy. And we wondered whether CB could be applicable to other proteins that undergo conformational change as well. So we started looking at published deep mutational scanning data sets on proteins that switch conformation to see whether we had any ability to predict the results of those DMS studies. So KRAS is a famous conformation switching protein, right? So it switches between this inactive and active form. And the active form is what specifically binds to downstream effectors and signals for cell growth and so on. There's a synthetic binder that is specific for the inactive conformation. And in this recent study, they did a DMS scan, so analyzing some 30,000 single and double mutants of KRAS, and they measured the binding against all of these effectors. And so what we wondered is whether CB scoring by kind of nominating certain KRAS variants as being open biased or closed biased could actually predict some of this binding phenotypes that were measured, right, in the study. So what we did is CB scoring of all of these KRAS variants, and then we plotted here on this plot, right? So based on the protein MPNN score against the active conformation versus the inactive conformation. And then in this plot, we colored the dots based on the experimentally measured binding to an active conformation specific effector, which is a RAL-GDS. And what you see is that the strongest binders to RAL-GDS are largely below the diagonal here, right? So indicating that the CB bias score has some ability to enrich the better binders to RAL-GDS. If we take the same exact scatter plot, but now we color based on the experimentally measured binding score to DARPin K27, right, which is specific for the inactive conformation, we see the reverse trend, right, with red kind of above the diagonal. And then this is just more quantitative violin plots where for every effector, we can look at the distribution of the binding scores for the top CB predicted active biased mutants versus the 2.5% most inactive biased mutants, and we can see significant differences between their measured binding scores, all in this direction, except for the DARPin, which is in the opposite direction. Okay, and we did this also for a deep mutational scanning data set on SARS-CoV-2 spike protein, and we could predict variants that were more biased to the up conformation, and those also experimentally showed stronger binding to ACE2, right, which binds only to the up conformation and not down conformation. So we were able to see signal on this system as well, and 7 other deep mutational scanning data sets. In fact, there were no deep mutational scanning data sets that we tried where we didn't see signal. So even though it's such a simple method, it really was able to enrich conformation-specific functionality across diverse protein types, so enzymes, a GPCR, synthetic binding pairs from Chris Garcia's group, and a transporter. Okay, this work is pre-printed, and Andrew has made this GitHub, which is kind of just a wrapper for protein MPNN, but the contrastive scoring, and would love for people to try it out. Okay, so back to FlexID. Okay, so we used conformational biasing, more ESM, predicted mutations, and also 3 mutations that we previously had picked up from directed evolution for another purpose that we know stabilize this enzyme. We mix and match them, and we end up with our final enzyme that has 9 mutations relative to template. We showed that this enzyme is useful, right? So this is a gel showing a whole bunch of endogenous proteins that are tagged by FlexID in different subcellular compartments where we genetically target the FlexID enzyme to the mitochondria, to the ER, to the ER lumen, to the cytosol, to the nucleus. And in each case, we can biotinylate like a whole bunch of endogenous proteins, enabling their detection and their enrichment. This is the most interesting part of this kind of the technology characterization to us. We were not expecting this when we started this out, but this is like a serendipitous discovery that has kind of unearthed, all these interesting questions about the mechanism. But we did a side-by-side comparison of FlexID to Apex and Turbo, which are very widely used, and we found that FlexID was the most spatially specific. We had assumed that, when we started engineering from the LPLA template, that it would catalyze promiscuous labeling in the same way as these other enzymes by generating a reactive species and then releasing it. But you can see, based on my kind of working model for how conformation regulates enzyme specificity, it's not a release mechanism. It's a contact-dependent yet promiscuous mechanism, right? The adenylate ester remains bound inside the enzyme. Okay, so we've been trying to explore this mechanistically, and I won't get into the details of it. I'll just summarize. Okay, so with TurboID and Apex, they're promiscuous because it generates. So TurboID generates an AMP ester and then releases it, and that kind of creates this cloud around the enzyme that can tag amine-containing protein targets. But remember, for FlexID, we think the mechanism of promiscuity is different. That it's not releasing the adenylate ester. The reason it's promiscuous is that it's basically removed this inhibition to the transfer step so that even bad substrates have reasonable affinity and can access that active site, right? So we've been doing some mechanistic enzymology here to understand this, and we have various lines of support that FlexID is indeed labeling by a fundamentally different mechanism than TurboID that doesn't involve release, that actually involves contact-dependent touching of endogenous proteins, but it's still promiscuous. Okay, so what are the lines of evidence? One is that we actually measure the off rate from FlexID, and it's very slow. We also know that the transfer step is enzyme catalyzed, whereas for Apex and for TurboID, that second step is not enzyme catalyzed. And then thirdly, FlexID actually shows some preference for sequence context that matches the electrostatic surface potential around the active site. Okay. So applications of FlexID that we're now exploring include proteomics in vivo, because it's non-toxic and because we don't have to worry about background from biotin anymore, right? So in vivo applications are facilitated. Because the labeling radius is now basically 0, right? Contact dependent, it's really spatially specific. We think it could be very useful for problems like discovery of receptors for orphan ligands, right? Where people have actually tried to solve that problem using tools like TurboID and Apex. It just, if you try to do that, you label the whole cell surface proteome, right? But if I really want to know what is exactly the cognate receptor for my ligand, and I don't want to label the neighborhood around it, I really need a very spatially specific enzyme, and I think FlexID might be able to do it. We've also developed a technology based on FlexID that I call PLF, for proximity labeling with fluorescence readout, instead of proximity labeling with like the typical mass spec-based enrichment readout. And we've used this as a high throughput screen for drugs, right? So it's basically a protein-protein interaction readout from living cells that you can scale up very readily. And so Shizong in the lab has collaborated with Nathaniel Gray's lab. It took him an hour to screen this library of 50-some molecular glues against 3 endogenous targets to see whether any of them are recruited to cereblon. So I'm excited about that application. Okay, so the summary of this part is that FlexID was engineered through protein MPNN-based conformational biasing and ESM. It's a fast, non-toxic enzyme that uses diverse non-biotin substrates, and it's more spatially specific than the existing enzymes due to this novel contact-dependent labeling mechanism that we could only discover by using these computational tools. So that's been really, really a fun adventure for us. And then we have this PLF method, and then conformational biasing with contrastive scoring using inverse folding models can predict conformational biasing mutations on a diverse set of natural proteins. It's super easy, super fast to implement, and when applied to LPLA, it drastically increased enzyme promiscuity by biasing to open conformation. Okay, yes, and it can be applied to other things. Now, the negatives. Okay, so here I just want to give some examples of where we've really tried to use computational methods and failed. Okay, just to highlight kind of as a working protein engineer where there are still a lot of unmet needs. Okay, so we're very interested in prediction of conformational ensembles. We have been using BioEmu since like January or something. We love it, and it's transformative for us. I am pleading with Frank to please make ligand-conditioned BioEmu. Let me show you this wild result. So FlexID, when it uses this BCN substrate, I already showed you, it's promiscuous, right? It catalyzes like, labeling of all these endogenous proteins. We see all these bands showing up. If we take that exact same enzyme and we give it a slightly different substrate, this transcyclooctene, it's active, but now it's specific. It's not promiscuous anymore, right? So our best guess is that with this ligand, it's open biased, but with this ligand, it's closed biased. So I want to understand that. We have no tools that predict this outcome. So I'm really interested in being able to predict conformational ensembles, but in a ligand-dependent way. Okay. Second wish. So we've been developing various technologies for antigen gating of receptors and enzymes. So this is our pager technology, and what it is a modular strategy for taking any GPCR and making it gated by any antigen that you choose, like whether soluble or cell attached, natural, unnatural, GFP, whatever. And the basic design is that we take our GPCR, we tether to it antagonist peptide that folds around and auto-inhibits it. But adjacent to that, we have a nanobody against our antigen. And antigen binding introduces a steric conflict with this peptide inhibitor such that inhibition is relieved, right? So basically, this is like a molecular switch that can either bind to antigen or it can auto-inhibit, but it can't do both at the same time. So in the presence of antigen, it's uninhibited, and then a natural or unnatural small molecule agonist can come in and turn it on. So this is a modular gating strategy. The bottleneck that prevents us from making pagers out of every single GPCR in the human genome is that we don't have peptide antagonists for most of them, right? So the ones that we demonstrated, we have like a dynorphin analog of the kappa opioid receptor. For the acetylcholine receptor-based GPCRs, we could use these snake venom toxins. Those worked. But for a lot of them that we're interested in, there's no known peptide inhibitor. Another example now, we're just trying to make antigen-gated turbo ID. So where it's auto-inhibited, and then in the presence of an antigen, inhibition is relieved, and then turbo ID can biotinylate stuff. Why do we want to do that? Well, I think it would be pretty transformative, because right now, if you want to do proximity labeling around a bait, you have to genetically fuse your turbo ID to it. But if you have a system like this, then I can do proximity labeling around endogenous targets, right? I just bring the nanobody to it, turbo ID turns on there. Any turbo ID that's not bound to your target is silenced, is off, right? So there's no off-target activity. Furthermore, I can then do proximity labeling around even non-protein things, like lipids or post-translationally modified forms of proteins, right? All that currently not possible. But I can't do this because I don't have a good peptide inhibitor. We've tried RF diffusion. We've tried pepperclip. Not doing it. So would love ideas for how to design better, more effective inhibitors for GPCRs and for enzymes. Okay. We create a lot of allosteric tools. Okay, so we're interested in taking enzymes, receptors, and being able to either regulate them with allosteric inputs, like light-sensitive domains, like the love domain, or calcium-sensitive domains, or have the conformational changes inherent to those systems, like a GPCR, allosterically regulate a fused enzyme, right? So for example, if we can take a GPCR, and we know this intracellular loop 3 is what changes the conformation the most upon GPCR activation. That's true across the whole GPCR family, right? That's how like GPCR-based biosensors by like Yulong Li are constructed, right? They put CP fluorescent protein here. So we want to put turbo ID there in such a way that it's inactive when the GPCR is off, but then when the GPCR turns on, there's a conformational change that somehow undistorts the turbo ID active site, so it turns on. And then if biotin is present, then it will generate a stable mark that we can read out by imaging or by Western blotting. So then we could turn any GPCR into an integrator, which is pretty useful. Right now people are making GPCR-based real-time biosensors, but being able to make integrators that give a stable, lasting mark that you can do fact sorting on, that you can do, imaging on, whatever, would be very useful. So this is a cartoon. So poor Gabriel, my postdoc, I asked him to make a schematic. He said he tried exhaustively to use AlphaFold, to use Chi, in complex with agonists and antagonists. He could not generate reasonable structures of the GPCR in an active versus inactive conformation, much less have any effect on the turbo ID that was fused to it. So we know experimentally that even tiny changes, like 1 or 2 amino acid, shorter, longer, change the amino acid identity in this linker region leads to dramatic changes in the allosteric coupling, right, between these two parts. I don't know of any computational method that can predict those results, but I would really love to have them. Okay, so we have some experimental data sets. They're not big, but they are high quality, right, where we've tested many variants that we've made by hand. and we know how, the degree of regulation and the direction of regulation. Okay, we also do a lot of tools that are based on splitting and circularly permuting proteins. Okay, so I know there are tools out there like Spell, we actually used before, for predicting good split sites. So if you're actually making tools that are based on split proteins, it's not just like, can you split it, and then can you get functional protein when you're done? No. So there's a lot more. There's a lot more that we need besides that. It depends on what your application is. Are you simply trying to make a mixing assay where if they're like anywhere near each other, then you reconstitute and then you have activity, right? Some applications are like that, like assays for endosomal escape into the cytosol. You just want a high affinity, flat fragments that if they're anywhere near each other, they just like zip up, okay? But maybe you want to report on proximity, right? Maybe you want to have a split system that reports whether A and B are actually brought together under native conditions. Then you have to have low affinity fragments that don't drive the interaction, but merely are passive reporters of the interaction, right? So can I have tools that help me design low affinity and high affinity split pairs? Furthermore, the fragments themselves have to be stable, folded, non-aggregating, right? Usually when you split, the resulting fragments, they're like hydrophobic guts, which are normally on the inside of the protein, are now like, flapping in the breeze. These proteins are not happy, right? So we also need, after they're brought together, very high reconstitution efficiency, right? So the activity has to be similar to the activity of the parental non-split enzyme. So humongous dynamic range. Circular permutation is basically the same thing as splitting, right? So you have to find some site that you're going to cut, okay? Make that the new NNC terminus, and then you have a long linker that joins the old NNC terminus together, right? We've done a lot of circular permutation. Most efforts to circularly permute a protein result in active protein, but the activity is usually very, very significantly compromised compared to the parental, right? And furthermore, usually we have like issues of proteolysis. Usually these linkers are very long, right? And they're disordered, and that introduces instability and sensitivity to proteases. So that's tricky. Okay, last one. So accurate prediction of protein binding modes and KD and KM of these modes. I had a chat with Yo this morning from Sergey's lab that was very helpful because I was wondering whether the Paraformer could help us with this. I guess one thing that's very difficult about the kind of problems that we're interested in here are that we have experimental data that our engineered proteins are interacting with native proteins, but we know all of those are never going to be found in the evolutionary record. Right? These are synthetic interactions that we've engineered. So computational methods that are based on evolutionary training are not going to be really helpful to us, right? But we're desperate to know, how are they interacting? What are their affinities, right? Or in the case of enzymes, what are the KM values? If you can help, let me know. Okay, so I want to wrap up by acknowledging my incredible team that has made all of this work possible. Peter and Andrew are just a dream team working together. It's been an incredible privilege to see their creativity and their hard work bringing all of this to fruition. And Shizang has been a close partner in all of the FlexID work as well as Albert has been helping with all these experiments too. And then we have amazing collaborators that allow us to do things like SACS and Wei and Christina are actually my former trainees, but we miss them, so we continue to collaborate with them on experiments. Our mass spec collaborator, our molecular glue collaborator, these are our other lab members, these are our sources of funding. Thank you for your attention.